So it's a lovely follow on actually from the last talk from Delaney and Dory, because my talk today it's basically a couple of updates or just features that weren't covered in detail last year, but it's also going to be a live workshop like we did last year. So if you would like to follow it, I suggest now you go to the polyploids.org website. You go to the workshops tab, which was already pointed out, and if you go for the very first link here in the top, the tools, you'll get the agenda. And if you follow the link on this session, which is polymapR and polyqtlR, you'll get the presentation, which is just a PowerPoint. But if you click on data sets, that will send you off to a Google Drive. And I suggest you download all now, okay? And then save that somewhere in a directory. So create a little folder somewhere, and if possible, start RStudio and set your working directory to that, or create a new project. I'll go back to that in a second. But anyway, I'm just saying this because downloading could take a while, especially if a lot of people click that button all at the same time. So I'm just seeing if we can get ahead of things. Okay, so my name is Peter Burke. I'm a researcher in Wageningen in the Department of Plant Breeding. And over the past few years, I've been working in polyploid genetics, developing tools, and of course, involved in this SCRI project. Now, I thought at the outset, it's a good idea to acknowledge people. Certainly, there's a big team of researchers in the Netherlands working on polyploids, in Wageningen in particular, Chris and Roeland, who were directly involved with the SCRI project, and also Paul Arends, who's one of the group leaders involved with the polyploids projects at Wageningen. I have to, of course, mention Christine Hackett, who was one of the co-developers or had a lot of input and inspiration in particularly polyqtlR. I spent some time working with her a couple of years ago in Scotland, so that was very useful. Today was supposed to be a live session, and Jonathan and Alejandro were supposed to be in San Diego, in sunny San Diego, right now helping out, but they did also offer a lot of help in going through the workshop files and suggesting improvements. So thanks to Jonathan and Alejandro for that. And of course, the TKI projects, they're sort of a Dutch public-private partnership between Wageningen and a lot of breeding companies. And so that's very much where the polymapR and polyqtlR tools came from. But in the past year or more, the support from the SCRI projects has also helped develop new features, and that's something I'm going to be talking about today. So that's the acknowledgements right at the start instead of the end. What am I going to be talking about now? Well, first of all, I'm going to give a very quick refresher on what is polymapR and polyqtlR and how does the pipeline look like. Then I'm going to talk about some updates or some new features for polymapR. That's the use of probabilistic genotypes. And in polyqtlR, I'm going to spend a bit more time. Going to look at cofactor fitting in an automated fashion to build a multi-QTL model. We're going to look at the topic of curating your genotype, so looking at errors and error curation. And finally, visualizing meiotic pairing patterns, so looking at whether your crop or your population is autopolyploid, allopolyploid, or something in between. So hopefully we'll get enough time today to go through that. Now it's going to be both static in the sense of PowerPoint, a couple of slides of me talking about it, but it's also going to be live, and that's why hopefully some of you have already launched RStudio. If you don't have RStudio, I'd suggest you download it now, but if I'm not sure if it's possible for you to get up and running if you still don't have RStudio on your computer to run with this live. But of course, you could be watching this back later on YouTube, in which case you have time to pause it and do that. Okay, so to start off with a bit of a refresher, first of all, of this pipeline, these two tools, polymapR and polyqtlR, they're separate OR packages, but they do talk to each other in the sense so that the output of polymapR is directly the input of polyqtlR. And they were developed really over a number of stages by basically needing tools at the time to do particular things. And that started really with work we were doing in potato. Potato is a very important crop, also in Wageningen, and plant breeding, very important, and also a very well-resourced crop. So a lot of things like snip arrays and so on, a lot of information. So it was a good crop to start on for developing these tools. At the same time, we were also developing a large axiom array for rows. And we had all this marker data from a tetraploid population. So we then used polymapR to map this rose, or polymapR developed in the mapping, shall we say, of this rose population, and allowed us also to deal with some non-random pairing in that population, so segmental allopolyploidy or allotetraploidy. At the same time, Geert van Geest was also doing his doctoral thesis work in chrysanthemum, which is a hexaploid, and so we needed to also consider hexaploidy in this tool. And we also had Arwa working in Alstroemeria, so in a triploid population. And that's another application that we needed to be able to deal with in the pipeline. Finally, when we had all that done, we decided diploids are probably important. And looking at the graph that Dave showed earlier, indeed, diploids are, after tetraploids, seemingly the most common crop among people involved in this polyploids project. And that was also something that we've said, well, it's low-hanging fruit, let's add that too. And more recently with the work of Alejandro working on strawberry, which is an allooctoploid, but as well, genetically speaking, behaves a bit more or less like a diploid, so a disomic species. So that's sort of where it came from. What are these tools doing? What do they do? So a very quick refresher, just a visual refresher of what polymapR does. It takes a set of markers and essentially goes through a number of steps to come up with a phased linkage map at the end. Now, these markers have been genotyped in some way, and that was in last year's workshop. Tools like polyrad, fitpoly, or updog, and so on. There's various, or supermassive, there's various tools for calling these markers and getting your genotypes. But once you have them, that's the input for polymapR. And there's a number of steps, for instance, identifying problematic markers and screen them out, and then going through a clustering, which also, in a sense, screens markers that don't cluster well. So your clustering is also a very important filtering or quality control step in the process. And once that's done, we use the MDS map package developed by Catherine Preedy and Chris Hackett in Dundee to do the marker ordering. So that gives you the order of the markers within these clusters, but it also gives you the distances between the markers. And finally, with the information that was gathered as such during the clustering and the linkage analysis, so the estimation of recombination frequency, the phasing is then done at the very end. So that's, and then once you have this phase map, this is essentially the key then to move on to QTL analysis using polyqtlR. So polyqtlR is a separate package. As I said, it takes the input as the phased map from polymapR as the input, and then it first estimates IBD probabilities using your known parental phasing and the marker dosages or marker scores. And once you have these IBDs and trait values, you make a sort of an association analysis and hopefully come to a point where you can find a QTL peak or a QTL position, and not only knowing the region, the locus of this QTL, but also the origin of the alleles that affect this QTL. So these parental homolog effects. So that's in a nutshell the polymapR and polyqtlR pipeline visually explained in a minute or two. Now, there have been some nice applications. As I said, in our group, we were working with a number of species, potato, rose, chrysanthemum, and so on. There have been applications by other groups, and I just wanted to highlight a couple of them that I've seen. I know Jason Zern and colleagues have been using polymapR in rose mapping applications. The most recent that I saw was published in 2020, where they used markers from a SNP array, an axiom array, to generate maps, but also to map a resistance to black spot disease in tetraploid rose. Another nice application in a completely different species is in an application in tetraploid kiwi. And kiwi is an extremely complex crop in the sense that it has not only polyploid, but has an enormous number of chromosomes. But polymapR was successfully used here also to generate maps and to map also a resistant locus there to Pseudomonas. And I think Gibran is also giving a talk directly after my workshop, so it'll be nice to hear also from Gibran again. Okay, so that's a number of applications. As I said, I'm going to talk here about some updates to polymapR. Now, there haven't been a huge number of updates, but probably the most significant update is, or a new feature to the package, is that it is now able to use probabilistic instead of discrete genotypes for doing the mapping. And what does this mean? Well, as I said, you start with your marker data, but that has been already called. You have used some prior step to assign scores to your individuals and your markers. So this genotype calling actually is a probabilistic process. And usually there's an extra step at the end to turn your probabilities back into zeros and ones and twos. So to discretize these probabilities. But in doing so, perhaps some information is lost. And so we thought, well, maybe we can also just adapt the pipeline to use these probabilities directly rather than having to go through this discretization step. So what do they look like? So you see here just a dummy sample data set of a number of SNPs, and these are the probabilities of each of these dosages. So the probability here of a 0 is 99 or 99%, and the probability of 1 is quite small, and so on. And in a discrete data set, that would be just scored here as a 0, and that would be a 1, and so on. But you could imagine there could be situations where you're not quite sure whether it's a 0 or a 1, and you might have a more intermediate probability. And in that case, usually a genotype caller would make that value missing. But by doing so, you're throwing away a certain amount of information that it is either a 0 or a 1. So there can be advantages to using these probabilities. So the polymapR pipeline has been adapted to that. And certainly I want to acknowledge here the input of Marcello Molinari in helping to implement some of the likelihood estimation part of that. So this is an example also of where polymapR and mappoly interact with each other in a sense. And Marcello was very helpful there. And it was published in the last year, and there's also a vignette on the CRAN website for polymapR now of how to use these probabilistic genotypes as well. And just to say that not only polymapR has been adapted, but also polyqtlR can use those probabilities as well as the genotypes. But so it's not just that you create your map and then you have to go back to having discrete dosages in polyqtlR. polyqtlR is also adapted for these types of probabilistic genotypes. That's all I'm going to say about that. If you're interested, either read the paper or have a look at the vignette. What I'm going to do now is switch over to the polyqtlR updates. There are 3 of them that I wanted to talk about today. Hopefully I'll have the time. The first is automatic cofactor fitting. Second is genotype curation, and third is visualization of meiosis and meiotic pairing patterns. And now we switch over to RStudio. So if you had downloaded this, so if you don't download these and save them to a folder, I've saved it in a folder called SCRI. I've also in the meantime launched RStudio and created a new project, and I set that project to an existing work directory. Again, these are simple steps, but just to remind you how to do it. So your working directory now should be that folder which has all these files, so the SCRI folder for myself here. If that's the case, then you should be able to open an R script file called the updates polyqtlR 2020. And here you see it. And this is actually just a purled version of the vignette. So the vignette is also one of these files. It's an HTML file. There, it has the exact same name. And if you open that, double click it, it should open in your browser. And let's see, you should be able to read that. So you can go through this at your own leisure, at your own pace, at home afterwards as well, if you're not following all the steps. Now, some of these steps do take quite a bit of time, like a number of minutes, and I'm joking about drinking coffee throughout it, but throughout this vignette. But for the sake of expediency, I've actually put those big files, usually they're actually IBD files usually, up on the Google Drive so that you can just skip over all those little moments of waiting. So that option is there. We'll see that in a minute. Well, the first thing to mention is that you need to install the polyqtlR package. And there has been an update. Just trying to think when the last update was. Probably about 3 or 4 months ago, maybe a bit more. I can't remember off the top of my head. I'm going to have to update it again because of this issue that's arisen. But the current version is version 007, just to say, and that's what is assumed in this workshop. But the new update to 008, which should be coming in the next week or two, won't affect any of these calls. Once you've installed the package, you call it into the working, so into this current session. And there's 3 files. And actually, if you notice, these are exactly the same files as we had last year. So I want this also to be very much a follow-on from last year's workshop. So it's the exact same phase map list file, the same set of SNPs, discrete SNPs for rows, tetraploid rows population, and also the same phenotype file. And this is not fake data. This is real SNP data from the Axiom array, as I said, in this tetraploid rows population. And these are real phenotypes measured over both Kenya and the Netherlands. So yeah, so we're not throwing completely perfect simulated data at you this morning or this afternoon. Now, the next thing that I do is I define something called NC. That's the number of cores. And well, on my computer, I've got 12 cores, so it's 11. I consider it to be the working number. A lot of these parallelizations are across linkage groups. And for those of you into rows, you probably know that rows has 7 chromosomes, seven linkage groups. So I'm actually not going to get any speed up over 7, but I'm going to leave it at 11 anyway, just because I'm a bit lazy. But yeah, so that's just to say a lot of these steps are optimized using parallelization. So that's what NC is. And as I said, I'm just going through basically all the steps that are described here in the vignette. Okay, so I'll just check the chat. So at the same time, I think Chris and I see Jonathan is replying to something maybe in the chat. So Chris should be there. Ruland is also maybe monitoring the chat. Jonathan maybe, and Alejandro is monitoring the chat. So I'm not going to be looking at that very much, but thanks to Jonathan. But if also feel free, we're a community here. So if you see a question that you can answer and you're not one of these people I just named, please do answer it. I'd really appreciate that as well. Okay, so let's get going. The first step is to estimate your IBD probabilities. And this is just a wrapper function. It has a number of options, a number of arguments, and I'm using the default settings on a lot of things. So the default setting, for instance, on the method that's been used is a hidden Markov model. And that's the model that was proposed and developed by Xiaozi Zheng in the tetraorigin package. And that was essentially brought into polyqtlR and then sort of expanded to deal with multiple ploidies. But basically, the description of the method is stemming from the work of Xiaozi and the development of tetraorigin. So you can see it's working here in the background. I've let bivalent decoding being false. So I'm telling the function, I want to also consider multivalence. I want to consider the possibility of double reduction. And I'm telling it that I want, well, I want to use parallel processing. So I'm specifying the n cores argument as well. It's a tetraploid, but the rest is fairly self-explanatory. The genotypes are these, and the phase map list is this phase map list that we just read in. Now it's finished executing, so I could visualize those IBDs. But one of the next steps, one of the steps that we often suggest to do is to spline those IBDs. So these probabilities were estimated at all our marker positions. So we could have 600 markers on a chromosome, we could have 800, or we could have large numbers. And of course, if we then move on to QTL analysis afterwards, we're going to have to do tests at all those separate marker positions. But there's a lot of redundancy in having that many markers. We don't need to do separate tests on all those marker positions. So it makes a lot of sense to interpolate or to create a more regular grid of positions with which to work. So the spline IBD function does that. The only thing is it takes a bit of time. And again, I'm a bit, I want to speed things up. So I've included on that Google Drive these spline IBDs as well already. So you can just directly read in. So you can skip over this step if you want, but it's good if you want to run it at home, just to see for yourself how long this takes. But it takes about as much time I've seen as estimating the IBDs themselves. But once you've done that, you don't have to do it again. It's going to be there, and you can save it afterwards as an RDS or whatever. The other thing we need to do is estimate blues, and we do that using the blue function, which is another function in the package. So just to say, all these functions are just functions part of the polyqtlR package. So by calling the library call at the start, you make all these functions available. If you get an error saying function not found or something, well, then maybe you probably just forgot to first load in all those functions with the library call. So we run the blue function, and that estimates these best linear unbiased estimates for, in this case, the number of prickles on the stem. So it's exactly the same as we had last year. And you see here, we get a data frame as a type of object in R, but it has two columns. On the one hand, our genotype names. So these are the individuals in that F1 population, their identifiers. And on the other column, we've got something called blue. So it's just giving it this name blue. And then these are essentially weighted means of your phenotype across these environments. Okay, once we have that, we're in a position then to do a QTL analysis. I mean, you don't have to estimate these blues, but it has an advantage when you're running a QTL analysis using the QTL scan function, and you particularly want to set a significance threshold. So you use the permutation test option being true. Well, then having these blues is much faster. The runtime is much smaller than if you had included the full experimental structure in this call. So it's recommended to first do the step of blues, and then afterwards to run your QTL scan. And you can see here it's already executed that, and it has it has the results here, and I've saved them in this object called QTL.ps for prickles on the stem. And you can see all the what's in that. There's various objects in that. I'm not going to go through that right now. But as I said, one of the updates that we introduced in the past year to the package was to automate the process of fitting cofactors. Now, if you remember last year's workshop, cofactor fitting for this particular trait was quite a big part of the QTL polyqtL workshop. And that was because there's actually some interesting interactions between those QTLs. So if you fit one of the QTL peaks as a cofactor, some interesting things start to happen at the other QTL peaks. But when we were getting this manuscript through the reviewing process, one of the reviewers said, and maybe you're out there, reviewer, it was a very nice suggestion, was could you automate this process? Could you make it more reproducible? So to satisfy that reviewer, we wrote a new function, check cofactors, which essentially goes through the process that I more or less did by hand manually in the workshop last year. Now, as I said, this is for the coffee drinkers among you. So it does take about 3 or 4 minutes to run this. And the reason being is that each time you fit a cofactor, a new significance threshold is estimated, it's re-estimated each time. The function basically goes through all the significant QTLs you found. It looks at all combinations of those and tests all different combinations of those QTLs as cofactors. It then sees are any new QTLs found or detected. And again, if new QTLs are detected, it goes through the whole process again. And each time it's trying to maximize a particular thing. And what it's trying to maximize is the difference between your QTL peak and your significance threshold. So what I call here is your delta LOD. So that's in a sense how high above a significance cutoff line your QTL peak gets. And so it's using this delta LOD as a way of trying to find the best position, or the most likely position for your QTL. Now, we noticed, and Jonathan and Alejandro pointed out, that when they ran it themselves, they got slightly different results than I did. And I think that's actually quite interesting and quite nice to point out, that this process of setting a significance threshold is not deterministic, because it's based on a permutation test. So each time you get a slightly different threshold. And because of that, then this delta LOD will be slightly different from run to run. But of course, we don't really care about the exact centimorgan position of a QTL. We're more interested in a confidence or a support interval around that QTL. So if the position is 13 centimorgans or 14 centimorgans, I'm not that worried. It's pointing to the exact same QTL. So for the purposes of this workshop, I think it's also informative that you understand that your results are not fixed, but qualitatively you should be getting the same results as what we see here in the vignette. So interesting for you also at home if you run it to see whether you get slightly different results to this. And the cofactor ID column, that's the column over here, that's telling you what were the cofactors involved. So where on what chromosome and on what centimorgan position, what were the cofactors involved that gave you this biggest contrast between your QTL, your LOD profile, your peak, and the threshold that was calculated for that particular QTL. And once you have those, so again, here's one I made earlier. We have this particular data frame. You can then pass that to the PVE function, and that's just a function for calculating the percentage of variance explained by your QTL model. And what it does in the case, there's 4 QTLs detected in this particular example. So it gives you the PVE for those 4 QTLs together, but it also looks at all QTL submodels, so all combinations of 3, 2, or the single QTL models. So that can be interesting as well to look at potential interactions between those QTLs and how they add up or not, as the case may be, to explain variance of your trait. Okay, now that's the first topic. I'm now going to change gears, as the saying goes, and look at something a bit different. Now, more and more people, I think, are moving away from SNP arrays and moving more and more towards sequence-based genotyping. And one of the consequences of this still is that you tend to have a higher error load in your data. And that can be very quickly seen if you create genetic maps, if you just look at them. So if you just plot them or just look at the table of their map lengths, you'll start to see there tends to be an inflation nowadays in the length of linkage maps. Traditionally, a map of about 100 centimorgans is the sort of the expected average length of a linkage map of a chromosome. And so if you start seeing map lengths of 300, 400 centimorgans, 500 centimorgans, then it's not that you've come across probably a groundbreaking scientific discovery that recombination has just gone off the charts, maybe due to, I don't know, COVID or something. No, it's probably more likely that your data has a lot of errors in it. And so one of the first ways you can see whether errors, genotyping errors, are an issue in your map is just look at the length of the map. And if they're above 100, 120 centimorgans in this case, there's probably an issue with errors. Now here, these are the rose maps that we produced for this tetraploid rose population. The map lengths are all, 80, 90 centimorgans. So there's not really an issue with genotyping errors in this data, but this is just demonstrating functionality. So it's not that there is a problem here. But suppose we were worried about genotyping errors. How else could we diagnose it in our data? Well, one of the things that I really like to do always is to, so first I load polymapR. Well, that's just so that you can plot this map. One of the things I always like to do is visualize your haplotypes, visualize these IBDs. So these are the IBDs that we estimated earlier, this blind IBDs in this case. And I'm using the visualize haplo function. This is this is in polyqtlR. To visualize those by name, I'm looking at linkage group 2, as if you can see here, linkage group 2, or I have it also right next to me here. Linkage group 2 was the longest linkage group. So if there's any issue with genotyping errors, perhaps it's on linkage group 2. And I'm going to pull out 9 offspring. I just do that by looking at the headers of the SNP dosages. So this is the row SNPs was of course the genotype data matrix. And the col names of that from 3 to 11 just give me the names of the first 9 individuals. So excluding the parents, which are in columns 1 and 2. And I tell the function, well, give me a grid of 3 times 3. And if I do that, you see here your first 9 individuals, and those blue bars represent the inherited parental homologue that individual carries. And you can see the recombination points here, or in this case, a whole homologue set was inherited altogether. But if we spot this particular individual, K007, if you've done this in your own data set and you see a lot of individuals like this, well, then there's probably something gone wrong with your genotyping, or maybe with your map construction. But you would like ideally to see these big, long, contiguous stretches of these IBDs. You wouldn't like to see all these stippled IBD profiles. But there is a way to solve. There's a way to get around that and to try to correct this issue. And that's what I'm going to show here. Now, one of the very powerful parts of using a hidden Markov model to estimate IBD probabilities is that you also can also model genotyping errors in that process. And more recently, actually, the work of Chao-Zi again in PolyOrigin has done a beautiful job of, shall we say, allowing local optimization of error probabilities within the hidden Markov model itself now, which gives you a specific error per marker, so per data point as such, per marker per individual, which is fantastic. But a sort of a workaround that's I would say quite robust and quite simple is just to run the process, run the IBD estimation multiple times at various different error priors, and then see which is the error prior that best fits your data. So again, I'm not going to run this because again, it'll take about 2 or 3 minutes, but there's the call itself, how you would do that. So I'm suggesting that these are plausible error priors here. So I'm saying, well, try 1% errors, try 2%, 5%, 10%, and 20% errors. So I'm giving the function sort of a set of errors that I think might be plausible. Anything higher than 0.2, 0.3 is probably unlikely. But we'll see in a second what happens. And again, I'll read in one that I calculated earlier. It takes a little bit of time to read in because it's quite a big file. And what is this object? Just multi. Let's run a bit fast. Let's have a look at that. So it's got 3 elements. This is a list with 3 elements. The first is an IBD list. So it's called here maximum likelihood IBDs. So this is essentially this first object here, this maxL IBD. That's the kind of thing that you would get as an output from your estimate IBD function. So this is exactly in the exact same format. So it itself is a list which has various objects in it. You can look at the documentation of the package to understand all the bells and whistles attached, but that's basically the big thing. And after that, you have 2 other objects. First is this vector of errors that we provided. So these are these numbers, 1, 2, and so on. And then the thing in the middle is these marginal likelihoods also returned. And that's quite useful for doing some plotting. Now there's a couple of lines of code here to produce some plots. I'll just go through them. I won't read through the code and explain this is what this part is doing and that's what that part is doing. You can go through it yourself. Of course, all this code you can use, but it's just a small loop to produce over the 7 rows linkage groups to produce these plots. What we're looking at here actually is these marginal likelihoods. And again, it was thanks to Chao-Zi in conversation with him that he pointed out that this is the best way to approach this issue. So again, thanks to Chao-Zi for that. You can see here, we've tried it at different error priors. And so the purple dots are at an error prior of 1%, so quite I would say a low error rate. And you can see across our individuals, so across the whole population here, and those purple dots tend to be the most likely. And these red dots, which is the much higher error prior, are much less likely, so they have a lower likelihood. So in all these individuals, more or less, it seems that an error prior of 1% is a good one. But there are a couple of exceptions. If we look here on linkage group 2, and you can maybe you can't see it very well here, but if you zoom in on the HTML yourself, you can see it. There's a couple of like an individual here where the order has been reversed. So in this individual, it's much more likely that there's an error prior of 10%. And that's interesting. You could ask yourself, is there a sort of systematics in those? Are those problematic individuals across the whole set of chromosomes? Or is it just something went wrong on linkage group 2? And something that I did show you already, I think last year, was, well, let's first of all pull out those individuals. How do we do that? Well, I'll just do this little pull out the names of which individuals have a likelihood less than -600, and these are log likelihoods on that linkage group. And that gives us the names of 3 individuals, and you'll see our individual 007. So the James Bond of this population is problematic in that linkage group. It has a likelihood, the most likely error rate there seems to be about 10%. So that's a bit of an issue. So we can look, are there systematics across all the chromosomes, and how do we do that? Let me see, I might be getting a bit ahead of myself on what's described in the vignette, and just see some other code here. Yeah, okay. So I think I'll swap over to the vignette for now, just to remind myself. So in the first instance, these are the 3 individuals above. So this is 007 and 24 and 88, which had these on linkage group 2, had an issue regarding their likelihood. And you can see, you can also see it seems to be a problem here. There's an awful lot of noise in these IBD profiles. What you see underneath that is the result of using this method, of increasing an error prior or testing different error priors and maximizing the likelihood. So selecting the error prior that maximizes that likelihood. Now it's still problematic, but I would argue less so. You can see there is certainly less evidence of all this stipple behavior. So, and of course, all this in reality would have to occur through all these sort of recombinations happening right next to each other. And so it does result in much more smooth IBD profiles. And that's something, yeah, that we want. And in another data set, I don't have it here, but you can also demonstrate, or I could demonstrate for you, the improvement that you get on a QTL analysis in such a problematic data set if you use this approach. What you tend to get is much smoother log profiles. So it does have an effect also on downstream applications, but right now we're dealing with the issue of genotype curation. So how do we see this systematics across the whole genome? Well, there's a function called count recombinations, pretty simple. And we'll apply this function to our IBD array, or to our IBD object. Again, here I'm pulling it out with the $ because it's a nested list, but you could of course assign this thing to something else called new IBDs, so that you don't always have to be typing the $ here. But what that means is I'm just pulling out this part of this list for those of you less familiar with OR. So this function just counts the recombinations for individuals and also across markers, and you can do some visualizations of the result. So here we see a sort of a distribution now of the counts of recombinations across all linkage groups across the whole genome, and there's a bit of a tail here where there's a number of individuals here that have a huge number of predicted recombinations, and this is even after we went through this process of, shall we say, smoothing out these IBDs or using a higher error prior where appropriate. And we can pull out those individuals. Let's see what are the names of those individuals in the tail. And there's 2 individuals involved, again, K88 and K24. What's interesting here, though, is that 007, so the James Bond of the Rose population, is not here. And I had a look why, and I could see with a little bit of looking around why that's not the case, but I'm going to leave that as an unanswered question right now. For those of you who would like to figure it out, I don't want to be giving away all the answers, but 007 is a problematic individual, but doesn't come out through this method of identifying problematic individuals. And I'm not going to give away the answer, but if you do, in a couple of hours, there's a sort of a, what do they call it, office hours. So it's during the lunch hour, actually, at 12 o'clock Pacific time. So I'd be interested if anybody had theories on why 007 was not found as having loads and loads of recombinations, even though we saw here earlier that it does. Anyway, that's a little challenge. I don't like to be patronizing people saying, here's homework for you. So if I sound patronizing, I apologize. Okay. Well, the whole crux of this work, or the whole point that we're driving at, is how can we use this to curate our genotypes? Can we do something about this issue? And of course, when we have these IBD profiles, I'll go back to here. When we have something looking like this here, this plot here actually gives us all the information we need to impute genotypes. I mean, we're seeing here at this position that we're carrying this homolog and this homolog and this homolog and this homolog in this individual. So if we have a marker here, we know what that marker score should be. You know, we could just apply. We've got our phase map. We've got everything we need just to figure out what's the marker score here. And similarly, across all the markers on our linkage group, we're able to use this profile to predict what our marker score is. And of course, for most markers, that'll be exactly the marker score that you had. But in these problematic individuals, or if you have a very problematic data set, if you pop up the error prior up high, smooth out your IBD probabilities, you can then just use that information to tell you what your marker score should have been. So you can use it for an imputation step. And there is a function for that. It was also there in last year's workshop, but I went through it quite quickly. So let's test how well this works. So there's a function called impute dosages. So first of all, there's a couple of lines of code here for how do you add errors to the data. As you saw, we don't have many errors in this data set. Sort of on average, there's sort of 1% errors predicted. So it's not a very problematic data set, but let's make problems. Let's add noise, let's add errors to this data. So I'm going to, these steps here, again, you don't have to worry too much about what I'm doing, but all I'm doing is I'm sampling a number of positions, 15% of the offspring positions, actually. I'm just randomly picking out from the whole genotype array, 15% of those positions. And I'm going to overwrite the scores there with new scores. So if it was a 0, I might change it to a 4. You know, if it was a 3, I'm going to change it to a 2. I'm going to add just to 15% of the data, complete rubbish, complete noise to the data. And that's in something called bad SNPs. So it looks exactly the same as your row SNPs matrix. So it's got the same format here. It's got your parents and your offspring. Now, notice I've only introduced errors in the offspring here. I've made it a little bit easy. Having issues with your parental scores is more problematic. And that's why, of course, we recommend that you genotype your parents more than once to make sure that they're well known and well genotypes. So you want to be sure about your parental scores if at all possible. But in the genotypes of the F1 population here, 15% of them are now wrong, or just errors, just completely wrong scores. Now, if we rerun this process, instead of using our row SNPs, if we use something else, if we use these bad SNPs as our genotypes, but for the rest, we do exactly the same as what we did before. Again, it'll take a coffee time amount of time to run, so I'm going to skip over it and read in the one I had earlier. But if we do this process, something interesting happens, which I think was very nice. And produce this next plot. So this is the same plot as we had before, showing these likelihoods of our individuals across these linkage groups. But what you'll start to see now is that the error prior of 1% are not likely anymore in comparison to these other error priors. In fact, it's the reds and the yellows which are competing for the top spot now. So the errors prior, so again, this is, when I say prior, it just means that we've told the function the probability of an error is 10%. So these error priors of 10 and 20% are much more likely now than those lower error priors that we had before. So a complete reversal of what we had in the previous step. So indeed, this hidden Markov model actually is able to correctly diagnose the rate of error in this data, which I introduced, which was 15%. And the final step then is to correct that. So use our new IBDs, the IBDs which in this case have got much higher error priors, to use those to smooth out these IBDs, but to use that to impute, the dosages that we would expect. Now, there are, again, a couple of arguments here. I'm not going to go into them right now, into the details. You can have a look at the documentation. And I'm going to call that thing imputed SNPs. And the function takes a couple of seconds, and it just gives you some output there that you can look at, telling you about missing value rates, about how many markers were able to be imputed, and the percentage of changes in across different, so for instance, 0 means no change. So 83% of the markers had no change, 6% of the markers had one change, and so on. And there was a certain, even 1% of the markers had up to 4 changes in those scores, in the scores across that marker. And the final, then the proof of the pudding, as it were, is to check what's the new error rate in our new kind of curated data set. So originally we had a 15% error rate, and in the curated data set, the imputed data set, it's gone back down to 1%. So we didn't quite correct absolutely everything, but we did pretty well. And we didn't also introduce a lot of missing values in the process. So with those new corrected genotypes, you could go back and redo a map. Of course, you could have removed those individuals that were problematic as well. And so there are some tools here that you could use for curating the maps. And this links nicely, I think, with the talk that's going to come up later. And that's the from Alejandro, looking at smooth as well, or smooth descent, which goes an extra step towards adding an additional element to being able to curate and improve your linkage maps in the presence of genotyping errors. So look out for that in a while. The very last point I want to talk about, and I'll switch back. I see I still have a couple of minutes. Should speed up a wee bit. Is visualization of meiotic patterns. And yeah, so there are some new functions in the package to do those visualizations using actually using visualizations out of iGraph. For those of you who know iGraph, it's another OR package. And so we see here these bit of code here, which is also in OR. I mean, it's in the OR file as such. That allows you to create this visualization of your pairing behavior, so your parental meiosis behavior. So deviations, so this is rose again. Rose is generally thought to be an autotetraploid, but of course it's a complex hybrid, so there's an awful lot of different species that went into producing modern roses. And it's a bit of a mystery why it behaves so much like an autotetraploid, because yeah, you would imagine that there's still some residual allopolyploidy in it, given the breeding history. And what we see here in this population, indeed, there are some deviations. So the thicker the line here between your nodes, and these nodes are your homologs. So there's 4 in I think this is parent 1 above and parent 2. And so here, for instance, a green line means an excess of pairing between these homologs. Now, this is a picture in rows, but what was interesting when we were publishing that manuscript on polyqtlR, we re-examined chrysanthemum data set, and this is a data set actually that's also publicly available with the publication from Geert van Geest. And in the original, one of the original applications of Geert was to figure out, is chrysanthemum an allopolyploid or is it an autopolyploid? Because there was a lot of, and apparently there still is some controversy in the literature about this question. And he concluded from just looking at markers on their own, so the segregation patterns of the markers individually, that it more or less showed hexasomic inheritance. So it looked like an auto hexaploid. But as I said, that was doing single marker tests. Well, when you go through the process of estimating your IBDs using polyqtlR or one of the other packages, and then produce this visualization, something really nice popped out. And you can see here, for instance, on linkage group 1, that 2 of those parental homologs, J and H, seem to pair with each other an awful lot more than you would expect by chance. In fact, the deviation is 114 more meioses that you would expect just by chance, by random pairing. So that's quite unexpected, and one would wonder, why are these homologs always pairing and recombining with each other? Is there something there? What is the biology behind that? But what's also, I thought, was very nice to see was this sort of a complementary pattern between pairing. For instance, here on linkage group 3, again in parent 2, you can see I and G, H and L, and J and K seem to have an affinity for pairing more often than not with each other. So that's sort of reminiscent of an allopolyploid, but still with pairing across the other combinations as well. And you can see also that sort of complementary pattern emerging, for instance, on linkage group 6 and linkage group 9, and to an extent in linkage group 4. So these, I think, can uncover some really nice biology, and for a breeder can be also quite relevant, I think, in terms of trying to get traits or trying to get recombinations between specific parental homologs. I think we're going in that direction with these sorts of tools, and this just gives us an extra dimension to try to figure out those things. So that's what I want to present. I'm still in time, I think, just about. So I'd like to thank again Dave and Oscar and Delaney and all the rest, Jeff, from the SCRI project for giving me the chance to share these updates with you. And I hope you have hope you enjoy working with the tools. And if you have questions, of course, they can be here in the chat, or we'll be here again in a couple of hours' time for the office hours. And of course, you can always email me. I really like to get feedback on these tools, or if you have questions or if you have problems in your analysis. I'm a friendly guy. I'll answer my email. So please do. I really appreciate if you have questions, do reach out and get in touch.